ahead and actually start recording. So in case somebody wants to go back and look at this, that'll be there. All right, any questions on that part there, guys, before I take it away? Okay, cool. All right. Okay, so I'm just gonna go over just briefly what next week looks like, and then we'll jump right into the material for today. So here's our weekly overview page for next week. Okay, so week 12. I changed something just a little bit on right there for next week, Wednesday. We were supposed to have a recorded lecture, but it was one that I really like to do with you guys in person because there's a lot of clicker questions, and we were going to have a free day the following week, so I just moved it to the following week instead, so there won't be any there won't be a recorded class for Wednesday next week. So we'll just have the live class on Monday with the Master A&P due, and then we'll have the second upper extremity CAT quiz on Thursday, and then the exam on Friday. Okay, exam 5, and then I'll also do the review session on Wednesday during class time like I normally do. Okay, so that's what next week looks like. Any questions on any of that?
So we'll still have the option to have the study session overview before.
I'll do the studies. I'll do the study session, so it's optional whether you want to come or not, but it'll be during class time on Wednesday.
Thank you.
Mm-hmm. Yeah, so the no class just means no new material coming to you on Wednesday. Yeah. Other questions on any of that? All right. Okay, so today we're going to dive into membrane potentials and really spend a lot of time in talking about how action potentials work and some of the mechanisms throughout that. Okay. Okay. Yeah, I had one question on the chat. Let me just show the answer to that. Some people were asking how to sign up for office hours. So if you go to the campus page here, the very top overview there where it says course overview, or start here, click on the virtual office hours page, and there's a link right there. You just click on that. You can sign up for appointments in my calendar. I have some extra appointments right now because of advising, so some of them say Dr. Sawyer's office hours, some of them say fall advising. Feel free to sign up for either type of time slot. That's fine. Okay, so definitely make use of that next week if you want to meet and talk about anything before exam 5. Thanks for asking that question. All right, okay, so we're going to dive into talking about action potentials primarily. So let's start out with just some terminology here. Okay, so depolarization, hyperpolarization, and repolarization are things that we're going to need to be familiar with what they mean. So depolarization means when the membrane potential, remember that voltage difference from one side of the membrane to another in the cell, when that becomes less negative or positive, okay, or moving towards the positive direction, right? But it's just a lot of times it's termed less negative because it starts negative, right? Remember, we've got resting membrane potential, which is about -70 millivolts, and so when it moves up in the positive direction, that's called depolarization. When it moves down below or less negative than resting membrane potential, we call that hyperpolarization. And then when it's been depolarized and on its way back down towards resting membrane potential, that's called repolarization. Okay, so why don't you on your apps right now, go ahead and just send in the answer to that clicker question right there, which would be which one of those three would be hyperpolarization, A, B, or C. All right, give you about 10 more seconds to send that in. It's A, B, or C. Okay, I got 62 participants and 59 people responding. Anybody else want to send that in? Give you a couple seconds to do that. Okay, almost got everybody there. See what you guys said. So most of you are saying C. Okay, so let's take a look at this. So A is definitely depolarization, B is repolarization, and C is hyperpolarization. So C was correct. Okay, any questions on those before we keep moving? Okay. All right, ready to move on? Okay. All right, so this is a review question that had to do with the last set of notes that was posted for Wednesday. So go ahead and answer this question on your app. What's considered electrical current when we're dealing with the cell membrane? Excuse me. So as it changes in the concentration gradient of ions, that changes in electrical gradient of ions, flow of electrons over membrane, flow of ions over membrane, or flow of electrons through an axon. Okay, so we talked about wires, electrical wires in the walls of your house. That is electrons flowing through those in order to, that's what electricity or current is in that case. But what does it mean in terms of a cell membrane? Okay, a couple more people, go ahead and send it in. All right, gonna shut it off. Good. Okay, so not quite sure. That looks almost like a normal distribution there. If that C was just a little bit taller, we'd have a nice normal distribution, but not typically what you want when there's one right answer. But yeah, so this would be flow of ions over a membrane. Okay, that's really what we're talking about. We're talking about current, electrical current in the body is referring to flow of ions over a membrane. Okay, so we've got so ions are, and the typical ones that we're going to talk about are going to be sodium and potassium. Okay, there's other ones that can cause some of these changes too, but typically we're talking about sodium and potassium. And so when if you go back to the notes from before, we talked about there's the potential energy we measure in voltage, right? But then we have kinetic energy, which is the current in amperage, right? And so this is what we're talking about here is the current part. What is it actually called? What is it actually in the body when there's actual current or electrical flow in the body? And that is the flow of ions across a membrane. Okay, any questions on that before we keep going? That's really what we're talking about with depolarization, repolarization, hyperpolarization, is it's all about those ions flowing across a cell membrane. Okay, so there's two main types of potentials that we'll talk about today. The first one's called a graded potential, and a graded potential is brief and it's local, and it's a change in the membrane potential. Okay, so there's a bit of change in that electrical potential across the cell membrane, but it's smaller, it's short-lived, and it just stays right there. It doesn't spread, okay? So it could either be a hyperpolarization or a depolarization graded potential, okay? So what that means, this image down here is showing you examples of depolarization type. So we're starting down here at resting membrane potential. We have a little bit of a bump in the positive direction, right? But then as soon as it's over, it starts going down, okay? Until we hit another stimulus right there, then it can build on that one a little bit and keep building up, okay? So graded potentials can build on each other, okay? And if we get enough of these, this is eventually going to get us up here to where the action potential threshold would be, and then we could have an action potential, okay? We'll talk more about that on Monday. We'll spend more time with graded potentials on Monday, but it's just important to know that graded potentials are what eventually will start an action potential, okay? So we could also have a graded potential in the opposite direction. It could go down instead, right? And that would be the hyperpolarization type. That would be moving us away from having an action potential instead of moving us towards an action potential. Okay, so they spread slowly, they die out quickly, they can be summed. That's what we're seeing here, where they build on top of each other if another stimulus hits before the first one was able to completely die out. Okay, so the purpose of these is either to start action potentials, that would be the depolarizing type, okay, or to block action potentials, that would be the hyperpolarizing type. Okay, so we're going to get into those a little bit more on Monday, but any questions about those right now? Okay, so those are graded potentials. All right, so now an action potential is a long distance signal that travels throughout the nervous system, okay? Long distance, it's fast moving, it doesn't die out, it's going to go all the way to the end of the axon, no matter what, okay? And it continues on its own. Okay, we have something called the all or none principle with action potentials, meaning that once we hit that threshold for an action potential, it's going to happen. Okay, the action potential is going to occur. It's not, and then it's not that we can have one action potential that's taller than the other one or a stronger action potential. They're all the same. Okay, so there's no variation in the magnitude of an action potential. Okay, so one action potential is this tall, the next one's also going to be that tall, also that tall. There's no variation in how high of depolarization we get with action potentials, even though those are a little bit different in height. That's just because I can't draw very well, we should have the same magnitude of that wave each time, and they don't sum on each other. Okay, they don't add up. The ones next to each other don't add up like we'd get with graded potentials. Okay, so this is really the language of the nervous system. Okay, this is how the nervous system communicates with itself as well as with all tissues in the body that it controls. Okay. All right. Any questions there?
I have one question. I don't know if it makes sense.
Sure.
So is the stimulus happening in the brain and the action potential happening like wherever it's directed? Or are they happening in the same place?
It's a good question. It can happen, all the above can happen. Yeah. So let's say we've got, doing something like having, thinking a thought, right? You have to, when you think about something, that's going to create a bunch of action potentials in your brain. So there had to be a stimulus from one part of your brain that sent an action potential to another part of your brain that started an action potential there, right? So we've kind of we usually have one neuron interacting with another one, and that's kind of where that stimulus comes from. Or we could also have a neuron in the brain sending the signal down the spinal cord out to a muscle. And so then that neuron in the brain had to be stimulated by something, another neuron higher, but then the neuron that gets out to the muscle, that becomes the stimulus for that muscle, for the action potential to start on that muscle. So there's always going to be kind of some initial stimulus, and and how it initially starts, like way up in the more complex part of the brain, in the prefrontal cortex, where we have conscious thought or subconscious thought. I don't have a good explanation of exactly like how that begins, but when we go from neuron to neuron communication, the first one is the stimulator, the next one is the one receiving that stimulus and starting the action potential, but it would be the same thing at a target tissue like a muscle too.
So it kind of just depends on what action we're talking about.
Yeah, but there's always going to be a stimulus that is most likely coming from another neuron that stimulates that neuron that we're talking about that starts the action potential.
Okay, thank you.
Yeah. Mm-hmm. Okay, other questions on that? Okay. All right, so action potential, we've already named these different states here, right? Resting membrane, or resting state, depolarization, repolarization, and hyperpolarization, okay? So, but the question is, what causes those changes in membrane potential, right? How do we go from 1 to 2? How do we go from resting to depolarization? How do we go from depolarization to repolarization and down to hyperpolarization? Well, it's all about this. It's all about changing the permeability of the membrane to different ions. Okay, and that's really what this bottom graph is showing you here, is that you can see the purple line in the background still is the action potential, which is the voltage graph, but then we can see that little orange one, which is sodium permeability, right? So between 1 and 2, sodium permeability shot way up, right? So that means all of a sudden the membrane opened to sodium, okay? And then we can see between 2 and 3, what happens? Potassium permeability goes up, right? So that means now the membrane was open to potassium, okay? And even between 3 and 4, we still have an increased membrane permeability to potassium. And so, yeah, so we'll see how that applies to this. So what dictates these changes in membrane permeability? It's all about these voltage-gated ion channels. And that's where we're going to spend most of our time. Next is talking about these different voltage-gated ion channels, primarily the sodium and the potassium ones. So again, what that means is the protein bound to the membrane that is at normal resting membrane potential, it's closed, okay? But then if for some reason the electrical charge around that membrane changes, then that can change the shape of that protein to where it opens up and now allows for an ion to pass through. And that's what it means to be a voltage-gated ion channel, that the voltage actually changes the shape of that protein in such a way to open or close the gate. Okay. All right, so let's look at the two channels that we're going to talk about. So we got the sodium channel and the potassium channel, okay? So we've got a few blanks to fill in here. So blank channels have 1 gate, the other channels have 2 gates, okay? So potassium channels have 1 gate, sodium channels have 2 gates, okay? So this one's our potassium channel. This one's our sodium channel. Okay, so the gate that we're talking about is this guy right there. Okay, so that's the 1 gate. In the sodium one, we got one here, and then we got one here. We got two separate little structures in there that can both serve as gates to block the flow of sodium. Okay, so potassium channels have 1 gate, sodium channels have 2 gates. Potassium channels have 2 states, which is really just closed or open, okay? But then these sodium channels have 3 states, okay? And we can see the name of them here, right? So resting state, activated state here, and inactivated state. So those are the 3 different names of the states in the sodium channels. And you can see the difference, right? Resting state means that this big gate, so this one's called the activation gate. The bigger one is called the activation gate. The smaller one is called the inactivation gate. Okay, so we got the activation gate and the inactivation gate. And so you can see at resting state, the activation gate's closed, inactivation gate's open. Right, so closed, open. The activated state is both are open. And the inactivated state is that the activation gate is still open, but the inactivation gate is closed. Okay, so there's only one of those 3 states where sodium can actually pass through, right? That's the activated state. The other 2 states are closed, but just closed in different ways. Okay, so crucial that you can wrap your head around that right now. So questions on that before we keep going?
What's the difference in function in like between the inactivated state and the resting state?
Yeah, we'll get there. It's a fantastic question. I'm glad you're already wondering about that, but we'll get there in a minute. Okay.
Is this why there's more potassium that leaks out than sodium that leaks in?
No, actually the leakage is driven completely by leakage channels. So there's whole other proteins that are there just to allow some leakage, okay? And there just happens to be more potassium leak channels than there are sodium leak channels. And that's why there's more potassium leakage than there is sodium leakage.
Thank you.
Yeah, for sure. Other questions on that?
In the activated state for the sodium channel, is the bottom gate closed or open?
Good question. I didn't write that on there. They're both open. Yeah, they're both open. So that activation gate's open and the inactivation gate is also open. Good. Anything else? Okay, so we're going to watch a video on the action potentials, which is right from Master in AMP. So you can access it as well under the AMP Flicks. I think it's the physiology animations. So let's go ahead and play that one.
Neurons send signals over long distances by generating and
Give me a thumbs up if you guys can hear that.
Most action potentials originate near the axon hillock of the cell body.
Most people
in
said
the
thumbs
initial
up. Let me
segment
turn
of
it.
the axon. It then travels the entire length of the axon. A closer
Probably, I
look
always appreciate that. He's reminded me to record and stop recording. So but yeah, so you definitely want to memorize these steps. This is a key. This is probably the biggest process that we have as far as exam 5 goes. Okay, so the steps of an action potential. And so yeah, so make sure to refer back to this slide. But I'll go ahead and walk us through the next few slides, and then I'll have a slide in a minute that I send you guys out to breakout rooms to discuss in a minute. Okay, so let's start out with how do we start an action potential, right? And so again, it has to do with, if we're talking about start an action potential on a neuron, then we might have other neurons like this interacting with the neuron that we're talking about. Okay, so this, in this case, we could call this the motor neuron. This would be the one that would go and activate a muscle. Okay, but there had to be some higher part of the brain that told that motor neuron that it's time to have an action potential, right? So that's what these higher neurons would be doing. So we see where the axons of that neuron either go right to the dendrites of that neuron, or they go to the cell body of the neuron. Okay, that creates this. That creates the graded potential, right? So we get some little threshold. We get some little graded potentials. If we get enough of them in a row and we hit this threshold, then we're going to see our action potential. Okay, and there's a special part of the axon where this happens, and it's called the axon hillock. The axon hillock has an extremely high density of those sodium and potassium channels. Okay, so those are that's why it's a better place to start an action potential. Okay, so what receives the stimulus to start an action potential? It would be the dendrites and the soma of the neuron, okay, of the body. All right, so that's our starting point. Okay, so we've gotten enough graded potentials to reach our action potential. So now let's talk about each step. Okay, we'll kind of break it up as far as the two channels go. Okay, so we got our sodium channel first. All right, so we got two gates: the activation gate, the inactivation gate. And actually, I would like you guys to do this on your own. I'm just going to show you. So when I send you to breakout rooms, I'd like you to go through this and just fill in what's going on. So in resting membrane potential, what's the activation gate? Is it open or closed? Inactivation, open or closed? Okay, so just do that for each of those. All right, and then same thing with this slide, which walks you through the potassium gate. All right, and there's just one passing channel, but there's just one gate, so it's just open or closed there. Okay, and then when you get to this one, I'd like you to just do these three things. Which ions are flowing which direction in each state? Okay, so in each of the A, B, C, D, E there, do we have sodium going in or out? Do we have potassium going in or out? Okay, you can name each stage. And then what I want you to send in on the Clicker app is number 3 here, the correct order of events. So A, B, C, D, E is not the correct order, but it's something else using those 5 letters. Okay, so everybody know what we're doing? All right, I'm going to send you to breakout rooms. And so you're working on those 3 slides. And even if you go 1 slide past that, it just matches up you can match up the part of the graph with that too. So if you get to that part, that's good too. All right, here we go. Nicole, is that you on the phone number? All right, so everybody's coming back now. If you don't have the answer to the clicker question yet, that's okay. Don't worry. I will give you some more time to do that. You don't have to send anything in yet. I'll open that up in a sec. But let's walk through these couple slides here first, okay? So we've got, remember the two gates, the activation gate, the inactivation gate, during resting membrane potential. Let's just do it this way so you guys can interact a little bit. Give me a thumbs up for any of them that are open and a thumbs down that are closed. Okay, so this first one, resting membrane potential, activation gate, open or closed. Thumbs up for open, thumbs down for closed. Okay, good. Most people are saying closed. How about inactivation gate, open or closed? That one's open. Good. Yep. Okay, and then in the depolarized state, the only way that we're going to let sodium in is if they're both open, right? They both got to be open for that. Okay. Now, repolarization, activation gate, open or closed? Still open, right? Yeah, it's stuck in that open position. What about the inactivation gate? Yeah, closed. Good. So repolarization is closed just, and resting membrane potential is closed, but it's closed in different ways, right? This one's closed by the activation gate being closed. Repolarization is closed by the inactivation gate being closed. Okay, and then during hyperpolarization, this one's a little bit different, and also I'll help you with this one. The activation gate starts out being closed, and the inactivation gate's open, but, or sorry, that's actually what it gets to, okay? And really what the key point here is this hyperpolarization actually causes the gates to reset, okay? So when the channel is stuck like this in the inactivated state, like it is here, then we're in what's called a refractory period where we can't have another action potential. It doesn't matter what we do to that. If another depolarization comes and it hits that inactivated channel, it doesn't do anything to it. It's just stuck in that spot, okay? So in order to get it to reset, so we can be ready for another action potential, we have to get the membrane down into hyperpolarization, and it's that hyperpolarization that causes the activation gate to close and the inactivation gate to open, so we can get back to this state. And then we can be ready for another action potential, okay? And that's part of the why that you asked me about, Emmy, and I'll talk a little bit more about that why in a second. Okay. All right, questions on that one, that slide? Okay. All right, then, oops. All right, so next one. So now we got the potassium channel, resting membrane, potential. The potassium channel is obviously going to be closed. What about depolarization, open or closed? Yeah, still closed, right? Okay. And how about repolarization, open or closed? Now we're open, right? Okay, now we're open. And then hyperpolarization, this one's a little bit trickier. What would you guys say, open or closed? Yeah, we're kind of somewhere in between, because actually how it works is we start out with it being open because that's what causes the hyperpolarization. But then once it gets down to being hyperpolarized, then it closes. Okay, so it's open in the early part of hyperpolarization until we get down to hyperpolarization, and then it closes. It gets reset just like the sodium channel does, and then it closes. So it's kind of two answers to that one. Okay, questions on that one? All right, so so then the really key point here is which ions are flowing in which direction for each one of those. Okay, so so obviously with these we can see the ions. The blue ones are the potassium, and the purple ones are the sodium. Okay, so obviously here we've got potassium flowing, we've got potassium flowing, we got sodium flowing here, nothing here, and nothing here. Okay. So then if we're going to name each stage, yeah, this is where it gets a little bit trickier, but well, first of all, potassium is flowing in or out. That's an important part. So potassium is always going to be flowing out. Okay, and sodium is always going to be flowing in. We're talking about the cell, right? So it's flowing into the cell or out of the cell. Okay, so now, yeah, so if we go back to the previous slides, it would be a good way to tell, you know, which two states did we have where sodium or where potassium was flowing out, right? One of those was repolarization, right? One of the repolarization was caused by potassium flowing out. And then the second one was hyperpolarization, right? So A and B have to be either repolarization or hyperpolarization. And the only difference is right here, right? That's the only way we can tell the difference between the two. Okay. In one stage, the sodium channel is in its resting state. In the other stage, it's in its inactivated state. Okay. So it would be in the inactivated state during repolarization, right? So A's got to be repolarization. Okay. And then after enough potassium is leave, now we've gotten down to hyperpolarization, that resets that sodium channel, or sorry, that potassium channel and the sodium channel, but the sodium channel, and that's why B is hyperpolarization. Okay. And then the only state that sodium is moving in has to be depolarization, right? Because that's the only one where we see sodium moving. So C's got to be depolarization. Okay. And then between D and E, we've got the only real difference is this, right? Is this local potential. Okay. So that's just it shows that that's the very start of it, right? Because that's when we get our first stimulus to get that depolarization going. Okay, so we would call that, yeah, that's the beginning. We don't really necessarily. We'll call D the rest. Oops, we'll call D resting, and we'll call this like what I called it before on the past slide, which was the stimulus received. I before E except after C. Sorry. Okay. All right. Okay. So does that kind of make sense? I know this is one of the tougher processes of the whole semester. So if you don't 100% get it right now, you've just got to spend some time with it, and I would expect that. Okay. It doesn't necessarily click immediately. Okay. If we go to the next slide, and we'll go back and put those in the right order in a second, but now we can see we can see the graph part here too, okay? So yeah, so I'll go back. All right, so the right order here. So if we can start with resting. We can call that 1, go to the stimulus being received, okay? Then we would go to depolarization. Then we would go to repolarization. Then we would go to hyperpolarization. Okay, I mean that, you could start out with resting if you wanted to, or you could start out with stimulus received and then end with resting. Either one, it doesn't matter because it goes around in kind of a cycle, but that's the basic order there. All right, questions on that? Okay. All right, so do you need more time on this slide before we go to the next one? Anybody tell me if you need more time? Right here. Okay, got all this written down. Okay, then this next one would just be the matching. Okay, so when we first start seeing that stimulus, we see a little bit of a graded potential, right? So that's got to be E. Okay, depolarization we already talked about was C, so that's got to be that one. All right, when we start going down into repolarization, that was that first one of A. When we get down further into repolarization, moving towards hyperpolarization, and it doesn't match up perfectly here, to be honest, because it just talks about it a little bit differently than the way that we did. So we had A, so it would have to be B here, okay? And then, yeah, A, B, C, D. So we're just left with D, which is resting state. And yeah, so we see a little bit of hyperpolarization there. That's the only reason I was saying it doesn't totally match up perfectly, but we're still seeing mostly resting there. So that's why that one would be D. Okay. All right. Questions on that process so far? All right, so you can definitely go back and watch that video on mastering AMP, and that can be helpful, and just try to walk through that, try to get that process down, know what's happening in each gate, in each state, which ion is flowing in which direction, and to cause each of those changes in membrane potential. Okay, so the cause of C is sodium going out, or sorry, sodium going in. Okay, the cause of A and B are potassium going in, potassium going in, right? So those are key things to understand there. Repolarization and hyperpolarization are caused by potassium influx, or sorry, efflux. Wait, goodness gracious, you guys, sorry. That should not be E. Out. Potassium always goes out, sodium always goes in. Sorry about that. Potassium always flows in, sodium, or sorry, potassium. Gosh, I'm really killing that, you guys. I'm so sorry. Okay, sodium always is flowing in because we got a lot of sodium outside, so it's always going to rush in when it has a chance. Potassium is always going to flow out because there's a lot of it on the inside. It's going to rush out when it has a chance. Okay, so what? Potassium out, sodium in. Hopefully I didn't completely confuse you on that.
So does this relate to like tonicity and osmolarity and all that?
It does, yeah, it definitely does. I don't really have a need for you to kind of tie the two concepts together yet at this point. Next semester, when we get into the kidneys and we talk about, yeah, sodium and potassium are very important in terms of urine production and things like that. And so that's where the two will kind of come together. But as far as the electrical part of it that we're talking about here, we don't really ever connect those two dots too much. Yeah, it's a good question though. Okay, other questions on that? All right, okay, so let's talk through this question. What's one biological purpose of the two gates in the voltage gated sodium channel? Okay, and I'll just go ahead and walk us through this one because this one is kind of something that we haven't talked about, but the question is the way of teaching it here. So would it be to allow action potentials to be stronger, spread in both directions, or really the bottom line is this one right here? Okay, to make sure action potentials only move in one direction. Okay, so because the fact that the sodium channel has those two gates, that's what allows for a inactivated state, right? If it just had one gate, it would either be open or closed. That would be it. But since we have this second gate, we can close one and keep the other one open, and that puts it in a state where even if a depolarization hits it, it doesn't have any effect on it. So what that means is, because think about this, we have billions of these little proteins lining an axon, okay? So the next slide, here, I'll come back to this one, but this next slide shows it well. As one of them depolarizes, okay, and then it moves down to the next one, okay? The ones back here, so the best place to look at it is probably this third one, okay? This one is inactivated, while this one is activated, right? So the action potential can't spread backwards because it hits that one that's inactivated. Okay. Same thing here. We get one that's activated. These ones back here are inactivated, so it can't spread backwards. Okay. And it can only spread forwards, and that's what we want because it would make no sense to the brain if it was receiving signals from a motor neuron, right? And it would make no sense to the brain if there were signals going away from the brain on a sensory neuron, right? And it would just be a big waste of energy too, because this is very energy costly to be constantly doing this, right? So in order for us to keep action potentials moving in one direction, that's one of the main purposes of having this second gate in the sodium channel. Okay, questions on that? Okay. So this is kind of what we're just talking about, depolarization of neighboring membrane, and then it opens up to the sodium, or opens up voltage gated sodium channels, okay? And then again, what keeps the action potential from going backwards towards the cell body? It's this absolute refractory period, which is caused by that sodium channel inactivation. And so the sodium channel has to be reset before another action potential can occur. And remember that resetting occurs via hyperpolarization. It won't reset until we can get to that hyperpolarization phase. Okay. Give you a second to keep writing that down. Any questions on that? See a lot of you still writing. Give you another few seconds here. All right. Like most of you are looking up now. Okay. All right. So this is the slide we just kind of talked about. So we'll pass that. Okay. So what about that transmission of an action potential and what different factors dictate how fast it can transmit? Okay. So there's really two main factors that dictate it. Number one is how big the axon is. Okay. So the larger an axon is, the more surface area we have for sodium channels, and then the more water we have inside of that axon as well. And water is a good conductor of electricity. Okay, so we've got less resistance with the larger, with the larger axon, and just a lot more sodium channels with the larger axon. Okay, so the bigger those axons are, the faster these action potentials travel, and then also myelination makes a big difference. Okay, so those Schwann cells that cause myelination in the peripheral nervous system, and the oligodendrocytes that cause myelination in the central nervous system. If we have the more insulation from that we have, then the less leakage of electrical current, and the action potential can kind of just skip past those insulated parts and only needs to get recharged kind of between the parts that have the insulation. Okay, so we'll talk about that, how it applies to different nerve fiber types or neuron fiber types. Okay, we got really three main categories, either group A, group B, or group C. And there's really just large, medium, and small as far as diameter goes. Group A also has very thick myelination. Group B has kind of mid-range, light myelination, and group C has no myelination. Okay, so the speed of transmission is incredibly fast in those group A fibers, 150 meters per second, which is about 334 miles per hour. Okay, so these are going to be all of our motor neurons for the most part, and then also some sensory information coming from our joints and muscles. Okay, the group B ones, the medium diameter ones, those travel a little over 30 miles per hour. These are most of our autonomic and visceral sensory neurons. And then lastly, our group C fibers travel pretty slow, only a little over 2 miles per hour. These are our pain fibers. A lot of our touch receptors travel on this. Okay, and when we see an electron micrograph image of a nerve, you can see it contains all different sizes of neurons, right? So we got some huge ones right here, and then we got some little tiny guys over here, right? So a nerve will contain all different types of neuron fibers. So bottom line here is it's about size. The bigger it is, the faster it goes, and then it's about myelination. The more myelination we have, the faster that signal travels. Okay. Questions on that? All right. Okay, so application question I'd like you to think about a little bit, and then read it. Go ahead and take a guess on what you think. Potassium levels in the body are a very important indicator of neuron health. How would very high extracellular potassium affect neurons? So normally we have very low extracellular potassium. So what would happen if all of a sudden we had a lot of potassium out there? Which of those stages of the action potential would that mess up? Okay, and if you're not sure, just take a guess, and we'll kind of walk through it. And there probably are multiple implications of this. Okay, but I'll give you about 10 more seconds, then I'll tell you kind of the most obvious implication of it. All right, anybody else? Okay, good. We got a normal distribution again. Looking good. Um, so yeah. So one of the like the direct implications of it would be both of these, right? Because in order to repolarize and hyperpolarize, we need a gradient for potassium to go out, right? Because normally we have a lot of potassium in, not much potassium out of the cell. So if all of a sudden we put a bunch of potassium outside of the cell, we no longer have a gradient for that potassium to leave the cell, right? So if we don't have a gradient for potassium to leave the cell, then we could depolarize still with our sodium, but then what's going to get us back down into repolarization if potassium can't leave, right? If potassium can't leave, we can't repolarize, we can't hyperpolarize, and so that sodium channel gets stuck in that inactivated state, and we can't have another action potential. Okay, so both repolarization and hyperpolarization would be a lot harder because we don't have our gradient for potassium anymore. Okay, so I'll get to how that a clinical application of that in 1 second, but let's talk about a couple of clinical applications of these of sodium first. Okay, so all local anesthetics, like many of you have had, if you've had some teeth work done, or you've had some kind of a procedure where they've got to numb you up first, almost all of those drugs work by blocking voltage-gated sodium channels. That's what they are. They're voltage-gated sodium channel blockers. Okay, so lidocaine, Marcaine, an epidural that a woman would receive during delivery, those are all voltage-gated sodium channel blockers. Okay, so that little channel with 2 gates, these little drugs are designed to bind to that thing and not let it open up. Okay, so we can't have an action potential on those sensory neurons, and so we don't feel anything, right? The pain doesn't get up to our brain. Okay, another application that as far as transmission of the action potentials, MS, multiple sclerosis, is an autoimmune disorder that destroys myelin sheaths. Okay, so just another thing that really makes it hard for action potentials to travel, makes motor control very difficult, because now all of a sudden, instead of those motor neurons traveling at about 300 miles an hour, they've slowed down to maybe 200 miles an hour, and that's not fast enough to move accurately like our body needs to. Okay, and then the potassium one. So this gets back to that clicker question. If extracellular potassium gets too high, potassium doesn't exit the cell as easily, we can't repolarize, and if we can't reset those gates, we can't have action potentials, okay? This is exactly what happens during transient spinal cord injuries, okay? When the spinal cord gets damaged, gets smashed, a bunch of potassium leaks out, and it makes it so you get paralysis in that whole region. All the neurons in that region don't work. So you might have extra paralysis in the early parts of the spinal cord injury that will eventually heal up, just because it was caused by that extracellular potassium being leaked out, okay? But the other application is this. One of the primary drugs inside of a lethal injection is potassium, okay? So it's actually a lethal drug. If you take too much potassium, you can't contract muscle, you can't contract your diaphragm, can't contract your heart, because you can't reset those gates and you can't have an action potential. That's one of the primary things inside of that lethal injection cocktail. But the opposite is a problem too. If we have too low of potassium, this lowers our resting membrane potential, okay, because too much leakage comes out and it hyperpolarizes us, right? And so then it takes more of a depolarization, more of those graded depolarizations to get an action potential, and it can lead to weakness and paralysis. This is common in elderly people that just have a potassium deficient diet, and it can also be caused by excess vomiting in cases of bulimia and things like that, because you lose a lot of potassium when you vomit. All right, guys, we made it. 3 minutes late, but we made it. Any questions that I can answer on this? Anything I can help clear up?
Yeah, I had one quick question.
Yeah.
So on the speed of transmissions slide, so is it saying the larger the axon? No, the one before.
Oh, the one before it. Yeah.
Yeah. So is it saying that the larger the axon, the slower the transmission is?
No, the faster.
Okay.
Good question. Yeah. Good. Other questions? Anything else I can clear up? All right, guys.
Question about when it
Go
goes down, like hyperpolarizing. So when
Yeah.
it drops down really low.
Hmm.
So is that just by the release of more Na in that it will pop back up? That's the part I was a little I'm confused.
Like, because it goes down in hyperpolarization, but then it works its way
Yeah.
back up to resting membrane potential. Yeah, what gets it back to resting membrane potential is most likely just the sodium potassium pumps kind of doing their job, which kind of helps to reset it. But in general, it's also just a little bit of leakage between sodium and potassium too that will eventually offset that and just work its way back to normal resting membrane potential.
Okay.
Yeah.
Thanks.
But not something I usually ask a question about. It's not really considered one of the stages, but yeah, it's a good question though. Okay, anything else, guys? All right. Well, good to be with you guys today. Have a great weekend. Happy Halloween. We'll show you some pictures of the fam this after the weekend with everybody's costumes. I'm dressing up as coronavirus. Hopefully nobody's offended to that. I got a bike helmet. I put a bunch of golf tees sticking out of it. So, and then my wife is Dr. Fauci, so she's gonna be chasing me around. Yeah, that's our costume. But you'll see the kids too. They'll be really cute, of course. So, hope you guys have fun. Stay safe out there, and we'll see you guys next week.